Dragi kolegice i kolege, dobar dan još jednom. Malo smo dinamični ovaj mesec. Imamo nekako dva, nekako skoro povezana predavanja. Danas imamo priliku da ugostimo kolegu iz Slovenije. So today we will speak in English. Our colleague is Aleksandar Janev. Aleksandar Janev, he is a research fellow and teaching assistant at the Institute of Cell Biology, Faculty of Medicine, University of Ljubljana. His work focuses on developing innovative therapeutic approaches for bladder cancer, building on his doctoral research into the anti-tumor effects of the placental amniotic membrane. He is involved in national and international projects on perinatal tissue derivatives and is an active member of the European Association for Cancer Research, International Placenta Stem Cell Society, and several Slovenian scientific societies. Aleksandar is visiting our institute through research visit within Amniotic Care Project, led by our colleague Christina, and he's an external collaborator. And he also will have lecture on the workshop. So today he will talk with us. Please, Aleksandar.
Hvala vam na lepom uvodu i to da ste željeli da vam pokažem šta mi radimo. Zaradi jasnoće ja ću pričati na engleskom jeziku, so you can understand me better. So thank you for all the listeners through online platform, and thank to all the colleagues that are here in the library. Today I will talk about the effects of human amniotic membrane in bladder cancer with in relation to 2D and 3D models of bladder cancer. So how my presentation will go. Firstly, I will present our research problems, what we are facing and how we are planning to battle it. Then I will show you the experimental setup that we used for this project. Then I will focus on the results and in the end I will sum up all the conclusions and give some future perspective what we're planning to do in the near future. So the main research problem of our research group is bladder cancer. Bladder cancer is one of the most common types of cancer that affects the urinary tract. I mean, it's the most common one. According to the latest Globocan data from 2024, it is the 9th most common cancer worldwide. Epidemiological studies rank Slovenia as one of the 7 countries in the world with growing incidence. And today in the morning, I just looked where Serbia ranks, and it's even worse. It's the 5th most common cancer in Serbia, according to the 2022 Globocan data, which affects both males and females. So why is this bad? Even though 75% of all the patients are diagnosed with non-muscle invasive bladder cancer, this cancer has really high recurrence rate. That means that these patients in 50 to 70% have recurrent bladder cancers that need to be removed by a urologist. If left untreated, 25% of those cancers progress to muscle invasive bladder cancer, which has really, really poor prognosis, with 5-year survival being less than 50%. So that's why we want to develop some new therapeutic approaches to battle this problem. In the past few years, there has been talk about the use of human amniotic membrane to battle this issue. So what is human amniotic membrane? It is a tissue that surrounds the fetus during the pregnancy. It consists of 2 layers. It's a layer of human amniotic epithelial cells and the human amniotic stroma, which is then subdivided into several layers. But inside the stroma, we also find human amniotic mesenchymal stromal cells. So it has two types of cells, and it has been known, it has been used throughout the history for many different diseases, but mostly in the ophthalmology and for wound healing. So the first human amniotic membrane use, it's dated many centuries ago. So we know that it has different properties. The studies have shown that it has really low immunogenicity. It decreases scarring, promotes epithelialization. That's why it is really good for wound healing and for in ophthalmology medicine. Also, it has immunomodulatory effect and antimicrobial effect. In the last few years, let's say the last 2 decades, it has also been shown that it possesses anti-cancer effect. So the aim of our research group was to investigate the effect of preparations made from this human amniotic membrane on 2D and 3D in vitro models of bladder cancer. So firstly, yes, we need to prepare those preparations. We go to the hospital, and after informed consent, after cesarean section, we peel off the human amniotic membrane from the underlying tissue, from the underlying chorion and placenta. So then the bloody amniotic membrane, which has been compromised with blood, is washed in the sterile hood. So we get, so this is like the typical, really nice looking tissue of amniotic membrane. Then we cut it into small pieces, like 3 × 3 centimeters. We measure the volume of those pieces, and then we add appropriate culture medium depending on which type of cells we are working with. After adding media, we homogenize it with different homogenizers. Here it's depicted a smoothie maker that we use for our daily in life for making smoothies from fruits and vegetables, but in this case it really works good because it mechanistically homogenizes the tissue. The non-homogenized part is then we filtrate it, so the non-homogenized part stays at the top of the filter, but the homogenized part that we term human amniotic membrane homogenate, we use for our experiments. So what is a human amniotic membrane homogenate? It's a mix of all soluble factors, all extracellular matrix, dead cells, debris of cells, and everything. If we want to use just the clear part, so mainly the soluble factors, we can centrifuge it and then use this blue part that we termed human amniotic membrane extract. So we have homogeneity, that it's a mix of everything, and extract just the soluble part. So I will show you now our experimental setup. In 2D models, we are mainly using 3 main cell lines: muscle-invasive bladder cancer cell line, non-invasive papillary cell line, and as a control, we are using normally porcine urothelial cells. So those are secondary cultures from the bladders of porcine bladders. We are developing primary cultures, and then we are using secondary cultures because you cannot find like normal urothelial human tissue. There is no indication for removing a normal tissue for the surgeons. So as a replacement, you can use transfected cell lines, or we are using normal porcine urothelial cells that have over 99% match in the DNA as ours. So it's like the maximum compatibility with human tissue. I will show you results on wound healing assay, transfer invasion assay. We also did RNA sequencing and some validation of those studies. In 3D models, we use 2 approaches. We use ultra-low attachment plate approach. So we seed cells in ultra-low attachment plates. Over time, they form big spheroids. We also are doing the matrigel dome-based approach. So in the matrigel drop, we are seeding single cells that over time make smaller spheroids. I will show you some of our results on patient-derived explants. So patient with non-invasive bladder cancer that are getting their tumors removed, we can get a biopsy, cut it into small fragments, and then cultivate it in ultra-low attachment plates, and then or normal plates, and then we have patient-derived explant. I will show some results on that topic. And lastly, in this year, I will show you some preliminary results on the proteomics profiling of those preparations. So what kind of proteins are inside. So let me start with showing you how human amniotic membrane preparation reduce the migration rate of muscle invasive bladder cells. So as you can see in these videos up here, the migration rate is reduced, but not only the migration rate, but also the type of migration. So you can see the control cells are migrating collectively. There are many adhesions between the cells, while the cells treated with amniotic membrane and a homogenate and extract have individual cell migration. So there are many intercellular spaces between those cells. So not only the migration rate is reduced, but also the type of the migration. In papillary tumors, so benign tumors, cells that are derived from papilloma tumors, we can see that the migration is slower because those are benign cells, but also it's reduced in comparison with the control cells. So as you can see, the control cells are moving faster than the treated ones. But what we saw is that there are many lamellopodia and filopodia along the leading edge of those cells, while in the treated samples, the number of those decreases. So it suppresses the migration rate, but it also decreases the amount of lamellopodia along the leading edge. Fortunately, in normal porcine urothelial cells, there was no reducing of migration morphology or pattern of migration. So the cells just close the wound, which is really nice. So it's like a cancer-specific effect. Then we used the transfer invasion assay to assess the effect on the invasion of muscle invasive cells. And as you can see, the control cells invaded in much larger number than the cells which are treated with human amniotic membrane extract, and those cells that were treated with the homogenate were invaded even less. So there was a dose-dependent manner if we diluted the sample. So we were thinking, okay, the preparations decrease migration and invasion. What can be the cell biological mechanisms of action? So firstly, we went blindly. So we just tested some of the most common proteins that are implicated in migration. And one of those proteins is a focal adhesion kinase, is a tyrosine kinase that has a signaling function, and it also is linked to the migration and to the attachment of the cells to the plate. So here, during my PhD 4 years ago, together with Christina, we did this qPCR analysis, and we saw really those cells that were treated, both invasive cells and the papillary cells, had lower expression of PTK2. That's a gene that codes for focal adhesion kinase, while the normal porcine urothelial cells retained the expression. We also then confirm it with Western blots. So in the green part, you can see the focal adhesion kinase, the phosphorylated form, which is like the active form that triggers downstream molecules, and also two molecules, Rho GTPases. Those are molecules that are along the leading edge and transform the membrane while the cell is moving. So we can see that decrease in all four of the markers that we checked, as well is in the benign cells, but in the normal cells there was no deregulation, downregulation, but more upregulation. So we just confirmed our migration results. Then in the last year or two, we knew that it's not only we did some more experiments with AKT, mTOR signaling pathway, and we showed this, but we knew that there are many molecules and many different signaling pathways. That's why we did RNA sequencing after treatment with homogenate. And as you can see from this Venn diagram, so here are the papillary cells, here are the muscle invasive, and here are the normal cells. The upregulated genes are with green, with a red color, and downregulated are with blue color. There's many genes, number of genes that are deregulated in the cancer cells in comparison with normal porcine urothelial cells. Here, I don't know if you can see it, but there are over 2,000 or 3,000 genes that are that have adjusted p-value less than 0.5 and for change over 0.5. So, but yes, the number of those genes, as you can see in the normal cells, is much less. So we know for a fact that it targets the cancer cells. I will show you some of our results. So yes, it alters the gene expression. This is just a volcano plot of the same data in the previous slide. We can see by the PCA analysis that the groups, the homogenate treated one and the control group, are really different in their gene profile. Also shown by this heat map. So you can see that the control cells and the treated ones have different gene profile. Then we use some tools for analysis what kind of signaling pathways may be involved. So the downregulated genes were mainly linked with immune cell function and signaling pathways that control our immune system, like TNF alpha, IL interleukin 17, NF kappa beta, and stuff like that. So the cytokine profile was targeted. That's why in the we chose randomly 15 differentially expressed genes associated with immune response, some interleukins, chemokines, and then we did some validation experiments where we saw a really decrease in interleukin 6, interleukin 8, and some of the chemokines 10 and 11. Also, we confirmed it with ELISA on the protein level. So we really validated those results, and this is just some of the results. We have many more unpublished data. But yes, we can conclude that the key signaling pathways were altered in muscle-invasive bladder cancer cells. After our 2D approach, we started implementing 3D models of bladder cancer. So we, as I said previously, we used muscle-invasive cells and papillary cells. We, in ultra-low attachment plate, 7 days, we cultivated them. And as you can see, the control cells in the control spheroids look like this. But when we added human amniotic membrane homogenate for 24 hours in the same, so after forming and 72 hours after forming, you can see that the architecture is really disrupted. So the morphology of those cells is really different. So this arrow shows that the parts of the amniotic membrane, the extracellular matrix, attaches to the cancer cells and affects the architecture, both in muscle-invasive cells and in RT4 cells. Papillary cells. We just did some preliminary, our student did some preliminary results, and we saw that only the phosphorylated focal adhesion kinase is downregulated. We need to go more in depth what kind of so what can we conclude? There are different signaling pathways in cancer, in spheroids, and in 2D. So we need to do more on the molecular part, and we are planning to do it. Since our institute is primarily working with microscopy, different kinds of microscopies, and we saw that the amniotic membrane disrupts the homogeneity, disrupts the architecture. We wanted to see whether the soluble factors in the extract did something. So we developed approach that is called in toto confocal microscopy. So we can we clear the spheroids. We remove the lipid parts, so the light can pass through the whole spheroid, and we can image up to 600 microns in width and in the depth of the spheroid. I can show you now. So we can see the whole spheroid from top to bottom. This is a control spheroid. This is a protein that is N-cadherin protein that is connected in the cellular, in the adhesion of two cells. And as you can see, with this approach, we can image the spheroid from top to bottom. And when we treated with the extract, however, there was no disruption of architecture. So in comparison with the homogenate, with the treated one with the homogenate spheroid, we cannot use it for this approach because it's, as I show you, it's really disrupted, and this approach is really, let's say, sensitive. So we need to have like a compact tissue in order to use this approach. So even though the extra does not impact the architecture, with this approach, we are using it on the different other models now, but this was the starting point. We can really see, let's say, 215 microns of width of the spheroid, and like we can do different kinds of reconstruction. So we get a spatial organization. Let's say if we use some drugs that are labeled, we can see if they penetrate to the a necrotic core and stuff like that. So this is a really good approach that we're planning to do more in our upcoming work. We knew that the extract does something, maybe not in the already formed spheroids. So that's why we use the matrigel approach, where we see that single cells in the matrigel dome. And as you can see, those single cell over time, like 2 hours, they are starting to form a smaller spheroids, and then over time the size of those spheroids is much larger. But when we treat it with extract, as you can see, maybe here are some single cells. They connect with each other, but if we I don't know, focus on this spheroid, we can see that over time it just vanishes. So it disintegrates, not just this one, but also every one of those spheroids. And we also did some results, which I would, with some experiments. We dissociated those cells and then we plated them on 2D surfaces, and we saw that those cells are practically dead. So there was no attachment of those cells in comparison with the control. So we know that the extract impairs the formation and disintegration of RT4 spheroids. You may ask why we use now RT4 spheroids, because in T24 spheroids that were used in our previous experiment degraded the matrigel. They are muscle invasive cells that express MMPs and other proteases, so we cannot see this formation of single spheroids, but we see the cells that are migrating to the well plate. So our last results from the 3D. So we got, so this is how a tumor looks like, a non-invasive tumor that is on the top of the urothelial cells, of the epithelial cells of the bladder. So we get those tumors. And then it's even bigger one. This is just a small piece. And then if we leave it in the culture for 7 days and do some paraffin-embedded sectioning and hematoxylin eosin staining, we see a normal cancer, benign hyperplasia. So there are many different layers of those tumors. But the treated ones, as you can see, after 7 days, similarly to the spheroids, there is a attachment of the arrows of the parts of the homogenate, the extracellular part, to the cancer cells. And as you can see, the tumor volume as well is reduced. The morphology of the tumors is dissipated. So not only in 3D samples from cell lines, but also in patient-derived tumors. So I will conclude with our results on the proteomics profiling. So in this year, we wanted to see what kind of molecules are inside our preparations. So our homogenate, we centrifuged it, and if you remember the clear part, we termed it as extract. But then we also lysed the debris, and we termed the sample as H. So those are more structural proteins. This is like a soluble fraction. And then we did TMT labeling. So we digested all the proteins. We outsourced it, of course. In Heidelberg, in the proteomics department, they added labeled peptide mix, which attached to each of those fragments. And by the liquid chromatography with mass spectroscopy, we identified the molecules that are inside. But we focused on how much of those proteins are in those fractions. Not what kind of proteins, but how much, whether they differ in the structural part or in the soluble part. So I will show you this one. So we identified over 4,000 proteins in all the groups. And even though the proteome profile is similar between the soluble fraction and the structural fraction, we can see that there is a big difference in the protein abundance. So how many proteins are in those sections? So in the supernatant part and in the lysate part. And it can be seen on the volcano plot, of course. If we compare the soluble part versus the structural part, we can see that the structural part has many more proteins than the soluble part, or not many proteins. The concentration of those proteins is higher, which may explain why the homogenate has better anti-cancer effect than the extract. So through other studies, we and others have shown that the human amniotic membrane can be used as a delivery system. So you can impregnate it with different kinds of, let's say, antibiotics, and then give it, and they will attach to the cells and give those antibiotics. So antibiotics, it can be all other chemotherapeutics, etc. So it's a really good delivery system, and this result showed that yes, the fraction part has many more proteins, level of proteins. So what kind of proteins are upregulated in that structural fraction? So if we look for the gene ontology of the cellular component, we can see that many of those proteins are implicated in membrane trafficking. So in trafficking of vesicles inside the cells. Those are endoplasmic reticulum membrane, mitochondrial membrane, and stuff like that, secretory granule membrane. So those proteins that are connected with the intracellular transport, Golgi membrane. But also there are proteins that are in the focal adhesions, so the cell attachment to the plate, and also cell substrate junction, so many different adhesion molecules. But what is the molecular function of those proteins? So there are proteins with GTPase activity, so those are like the CDC42, RAS, C123. Those are proteins that affect the lamellopodia along the leading edge. Many cadherin binding proteins. Those are proteins that are cellular, in cellular adhesions, insulin-like growth factor binding, so proteins implicated in growth, and etc. So this is like some preliminary results. We need to go more deeper, and of course, we need to validate those results with different kinds of methods, but this is just like preliminary ongoing data. So with this, I would like to conclude, and I hope that I showed you that the preparations, both homogeneous and extract, have a very broad effect, and they affect the cells on 2D and 3D level. And using this transcriptomic, proteomic, and microscopy approaches, we really showed that this is a promising candidate for further animal studies. But better, now we are planning to to develop new co-culture systems where we will combine normal and urothelial cells to model the tumor microenvironment, also add some immune cells. And our ultimate goal is to prepare patient-derived organoids to better replicate the in vivo tissue architecture in order to use less animal models. And lastly, we need to dive deeper in the pathway discovery and translation. All the multi-omics approaches, we need to validate them and study them more. And with this, I would like to conclude. I would like to acknowledge all my co-workers, especially Professor Matej Kreft, which started this work together with Taja Železnik-Ramuta on the amniotic membrane. I would like to thank all our co-workers, especially our students that are doing the work, and our technical as technicians. Yes. Without them, this work would not be possible. Also, thanks to Christina for the invitation, and also tomorrow for the workshop. Please join us. I would like to thank the medical center for really having a nice relation, and we can call them in the morning and have the amniotic membrane in our lab in 2 hours after the call. So yes, thanks to them. And lastly, I'd like to thank our funding core. But I would like to thank all the mothers who donated placenta and make this research possible. Hvala vam na pažnji i ako imate pitanja, može.
Pričali smo na početku, možemo i na srpskom.
Možete da pitate,
Okay.
da.
Pričamo na srpskom, ovo nije moja tema, istraživanja, tako da preosnovno hvala. Bilo je divno, inspirativno i veoma značajno. Obzirom na situaciju da živimo u eri gde kancer predstavlja veliki problem generalno i stvarno je fenomenalno predavanje. Svaka čast. I nadam se da će to prevashodno prvo da se podigne, a i mogu to i da te pitam na viši nivo, neki ozbiljniji, neke predkliničke studije ili kako to već funkcioniše, tako da mislim da je vrlo veliki značaj i sad koji su ti naredni korak, naravno, osim ovoga što se priča, ali neki, da kažem, više ti značajniji koraci da se to malo prevede u taj nivo predklinički.
I will respond in English,
yes,
because I feel more confident.
Yes.
So the next steps would be to replicate the tumor microenvironment. I didn't show you, maybe now it's the time what my colleague is working on, but we can co-culture normal and cancer cells, and then we can study, like I wanted to show this presentation, but it didn't go with my amniotic membrane. It's not related, but we can, for example, here, we can capture the moments when the invading cells, those are the T24 cells with red, here with orange. So we can really, with this approach, we can really study what kind of molecules are there. So we can, or what types of signaling pathways, what triggers, for example, those cells are not invading now, but like those cells, those that are here, we can see the moment when, so we can really dive deeper in the signaling pathway. So different kind of co-culture systems, and then the organoids are the ultimate goal.
More methods. New.
Yes, because we need to translate our research into more clinically appropriate systems. Those are nice systems, but they do not replicate the in vivo. Yes.
Okay.
So that's one of the.
But it's great. Okay. Thank you. So please.
Mislim, imamo čas sjeti sutra da radijati malo, ukoliko ste nešto pitali. Šalim se, super je, mislim, ja sam već znala dosta rezultata, ali ovo novo, mislim da je fenomenalno i da će nam značiti za ovaj projekat. A moram da kažem da stvarno pohvalim Aleksana zato što sam pristrasna, ali mislim da je čitava priča amnijanske membrane došla ovde zahvaljujući Aleksu, Mi smo se upoznali na sastanku kosta akcije koji se bavila perinatalnim tkivima. Ja sam se tad bavila ćelijama pupčanika, Aleks se već uveliko bavio amnionskom membranom. Tada smo se upoznali, to je dovelo do moje posjete njima, do njegove posjete ovdje i do mog postdoka tamo, da bismo onda amnionsku membranu doveli ovdje s ovim projektom, s Ivaninom bilateralom, tako da mislim da je on zaslužen za stvarno mnoge stvari ovdje i pored toga je moj neki utisak. Mislim, i nije neka velika razlika u godinama, ne želim da pričam sa neke pozicije nekog koji je tad već doktorirao, nego stvarno kao ono što bi se reklo lab mate, da je Aleks jako dobar naučnik u smislu da mozak mu radi jako brzo mu padaju na pamet nove ideje, jako se brzo snalazi i u situaciji kad je meni tamo isto nešto bilo novo, njemu je to jako je proaktivan, jako je pametan, jako je dobar saradnik u labu, a meni na kraju stalno postao prijatelj, ali to su sad neke druge teme, u svakom slučaju Aleks, drago mi što sad odvojio vreme iz ovo, jeste malo bilo brzo, da smo te to pitali pred kraj, i nisam ni očekivala da će rezultati biti ovako dobri i da ćeš imati neke nove analize koje bar nam ovdje dali stvarno high-tech.
Hvala na lepim rečima, ne znam šta da kažem, ali ja sam ovdje, ja bih želio ako neko zna da kažemo proteomiku i transkriptomiku, da malo popričamo, da nešto novo naučim, jer to je područje koje mislim da je sad baš in i to je budućnost istraživanja, iako smo mi, sad ako me čuju, nadam se da nisu moje kolege koji su primarno mikroskopisti, a kako se kaže, koji se bave mikroskopiranjem, ta mikroskopija nam da vidimo stvari, ali takve male detalje možemo samo sa ovim pristupom transkriptomike, omike, protomike, tako da ako neko znam, ali bilo što, kakav komentar ili možda savjet, ja sam tu i ja bih želeo da popričam.
Kada je pominjala koordinatu i to sasvim logičan korak. Kako ste došli na ideju da baš analizirate te dve grupe koje ste uzeli? Smatranje, naravno, to je potpuno smisleno.
Zato što znamo da je, because we know that the homogeneity is a mixture of everything, so in order to capture everything, we need to remove the soluble part, but also we need to check. So we know what amniotic membrane has. It has been shown what kind of molecule, like if you remove the tissue itself and lyophilize it and do proteomics, you know what kind of molecules. But with this homogenation, it's a mechanical disruption of everything. So yes, everything, the cells are not viable. Maybe some of those are which were not impacted, and that's why. We wanted just to check what kind of, not what kind, but also what kind. We also did some different DII approach of transcriptomics that I did not show here, so we know how many, what kind of molecules. Here for this approach, we just wanted to show the lysate part really has more proteins, and maybe that's why it has greater effect, because if we compare all of our studies with homogeneity in the extract, the homogeneity is superior. I mean, the extract has anti-cancer effect, but when those molecules attach to the cancer cells, it really brings all those stuff that are attached directly to the cancer cells. So our idea is maybe use it as tomorrow as some studies to add the chemotherapeutics in the amniotic membrane and then give it to the cancer cells. Because the disadvantage of chemotherapeutics is that it also affects the normal cells, right? Everything. But if we target the delivery, that would be good. And for the proteomics, it's just the starting point. If you have ideas, which software to use, because we do not have bioinformaticians, everything that I show, it's by myself. So I'm like at the lowest point of the on YouTube videos and stuff like that. How to, if you know some new approaches, I would be glad to hear them.
Mislim,
Do you think that maybe some effects which you saw from amniotic membrane homogeneity comes from just physical
Yes.
interaction
Yes.
particles? And did you measure the size of these particles?
Yes.
Yes.
I mean, this is a scanning electron microscopy picture. It's in Slovenian, but it's the same. I knew that this question. This was my first question when I started it. So the lipid membrane, a plasma membrane, is very different in cancer cells and in normal cells. My colleagues already showed this one. And we know that if you, let's say, if me and my colleague wash the cells and my force of washing is slower than yours, more cells will detach when you wash them down. So there is 100% mechanical reasoning behind this. 100%, I'm sure of it. But this is this can be good because here in this you can see the so the attachment of the extracellular matrix just on top of the cancer and muscle invasive and non-muscle invasive, but not on the normal cells. So definitely there is. That's why we use the extract. So extract, it's a clear supernatant. It also has effect, but not as much as the.
But for the application, would you choose extract or
For microscopy reasons, I would choose the extract because the debris, I mean, okay, now we have our confocal microscope that we can, which is inverted, so the parts do not affect our image. But on the other hand, and I would use extract, but on the other hand, the effect also antimicrobial immunomodulatory is better with the homogeneous. So I don't know. I want to find the proteins that are inhomogeneous and they maybe add them in some in some let's say PBS or something, and then try it. Like why? Because of maybe is this the molecule that is affecting the cells and stuff like that. So this is the ultimate goal, but I don't know whether it's feasible. 2 months. 2 months. Yes, we got the results in no, not 2 months. In May. So it's of course. Yes, that would be ideal.
I can share one contact with you. Expert protein.
Or yeah.
He's also expert in cancer.
Oh, that's really nice. Yeah, thank you. That would be appreciated. Thank you.
Ok, to je sve za danas. Još jednom, Aleksandar, hvala puno.
Hvala vam.
Predavanje je stvarno divno, inspirativno i nadam se da ćete za neko kratko vreme sad uspjeti da napravite neki ozbiljni sljedeći pomak u tom nekom finalnom cilju, da to sve uđe u neka predklinička i klinička istraživanja. To je stvarno bitno za sve nas. Hvala ti još jednom.
Hvala na pozivu, hvala svima.
Znate, testina već zatvorena za nove polaznike, ako neko će se prijavi, ali u svakom slučaju želim vam lepo druženje, dosta da učite i da ne bude jedino, samo prvo u nizu. Toplo se nadamo tome. Još jednom hvala Aleksandra i dobrodošli ste ponovo.
Hvala vam, hvala na pozivu.